Hello everyone, thanks so much for sitting today here and listening my talk. Let me introduce myself. I'm Xavier Benito. I'm a Beatriz de Pinós postdoctoral fellow at IRTA, and my research revolves around understanding long-term environmental change, applying multivariate time series analysis, using different type of proxies to study aquatic community resilience. I'm really thankful to the organizers for inviting me and give a keynote talk in this first ever virtual international Diatom Symposium. Today, I'll be talking about shifts, baselines, and non-linearities in order to quantify ecological resilience through lens of long-term limnology, and of course, I'll be using diatoms, and more specifically, our tropical South American diatom dataset. The world is changing at fast rates and magnitudes. No big news here. Everyone knows it. So this is the famous hockey sticks plot on rapid change in the Anthropocene by William Steffen and collaborators. As you can see, there exists a wide array of tip changes in drivers in terrestrial, freshwater, marine, and sociological systems in the 1800s. Steep changes include both socioeconomic and earth components, such as water use, construction of large dams, ocean acidification, and loss of tropical forests. So we have global problems that require global solutions. Different systems such as lakes, rangelands, and coral reefs are documented to experience large and abrupt shifts in its function and structure following these steep changes in drivers. These abrupt shifts are often regarded as regime shifts. Some include desertifications in rangelands, coral reef degradation, and also eutrophication in lakes. In all these cases, the theory of resilience predicts that for the same level of disturbance, let's say total phosphorus in lakes, two totally different states in the system are possible. For instance, in lakes, we might have a state dominated by macrophytes, so we have clear waters, versus a state dominated by phytoplankton, having therefore turbid waters. Anticipating these abrupt changes and recovering from them is a major practical and theoretical challenge, particularly given the long lags between often some remedial actions that managers do and the ecosystem responses that occur when undesirable states are themselves resilient. So we need to understand not only why, but when these changes occur, because it's crucial for ecosystem functioning and also managing better ecosystem services that lakes in particular provide. In the last year, different modeling studies have been looking at when rates of change, abrupt rate of change of a species may be exposed to unprecedented environmental conditions. So this is one of the recent studies by Chris Thiesos and collaborators using future climate model ensembles, the RCP 8.5, and they are looking at when species will be falling outside their thermal niche. And as you can see, for terrestrial assemblages, almost 90% of the ecosystems will have at least one species outside their thermal niche by the end of the century, and this pattern is strong in the tropical regions. One of the reasons of this is because tropical and equatorial areas have a narrow historical climate variability and stability, so any impacts that the region will have might have impending impacts on species and ecosystems. In a very recent study, Juan Rocha, using statistical indicators of resilience loss, such as increase in variance, standard deviation, or an autocorrelation, also pointed out that primary production will be losing their structure and crossing critical thresholds in tropical regions, reinforcing this area therefore as areas that are at high risk, suffering critical transitions. Regime shifts can be interpreted in terms of resilience. This is here I'm showing you two cartoons of the ball and cap analogy. On the left, we have a high resilient state, which is depicted by a deep basin of attraction, and in which the ecosystem depicted by the ball, every time that suffers a perturbation, it recovers rapidly to an equilibrium. On the right hand, we have a low resilient state characterized by a much shallower basin of attraction, and ecosystems suffering disturbance are more prone to cross a critical transition and entering into an alternative stable state. This theory has been widely developed and mostly modeled using monitoring data, observational datasets, long-term datasets, but exactly the same theory or reasoning can also be applied using long-term ecological datasets and proxy data. This is how I envision this theory using paleolimnological data, not only one basin of one alternative stable state, but also two can be reconstructed using this type of long-term data, and also having a wide catalog of driver and ecological responses in order to understand better processes and dynamics, and at the end having a catalog of which perturbances are worthwhile to tackle, and at some point reduce or restore critical drivers. So coming along as well on this importance why long-term records are important, I would like to in the next couple of slides, using our own research, showing you the importance of long-term ecological data, exemplifying two concepts that are widely embedded into our literature. The first one is the invisible present, which question whether unusual events are truly unusual or are an artifact of the observational approach and sometimes the perspective of the investigators. The shifting baseline concept, on the other hand, questions the situation in which the state of our natural environment is lost over time, and hence every new generation accepts certain levels of degradation because they don't really perceive that changes that are actually happening. So long-term data and paleolimnological observations can help us in tackle these two questions. For those that are not familiar with the paleolimnological approach, in this plot I'm trying to summarize the methods and some modifications that I'll be talking about later in this talk. So the way how lake sediments can be used to reconstruct past environmental changes are first developed using what we called a biological training set, of course, talking about Diatom training set, in which we linked species distribution and abundances with environmental data, those ecological variables that are supposed to have an ecological effect, a direct and sensitive ecological effect on a species, to construct a kind of regression model to later can be used in time down-core limnological observations to reconstruct paleolimnological conditions. But in this case, what I'll be doing and show you guys later a couple of examples is that using this Diatom training set, we can reconstruct also those variables that are also measured on the field and extract inferred values using a multivariate ordination method. Then using this would be the spatial approach, or space for time substitution if you like. But then if we have Diatom sequence and proxy data recovered from sediment cores, such as XRF data, isotopic composition of the sediments, we can also do exactly the same, infer the values using a multivariate ordination technique coupled with regression methods, and at the end coming up with this coupling fit that I defined, which we have the spatial trend of the variable and the temporal trend of the variable using these two complementary methods, and at the end calculate the coupling fit to see to what extent these expectations from reference set of sites estimated independently fit with their proxy data in the sediment record. Then we can start answer questions or posing questions about lags, equilibrium, or alternative states. I'll be coming back to this method later with a couple of examples. Here I'm showing you how different types of diatom community trajectories over centennial and millennial time scales can be unfolding in high elevation lakes of tropical South America. For instance, this is a nice example of how the invisible present is exemplified when at short time scales abrupt change in the plant thick diatom discostalis teligera can be defined or characterized, whereas at longer time scales populations may follow more gradual changes. If we can recover longer sediment records, or even the species, this type of the species might be following periods in which they were extirpated from the sediment record, and then they recover, even being one of the most significant species, abundant species in the record. So we wanted to kind of model and categorize, coming up with a framework using 4 Ecuadorian lakes located in the high Andes and Interandean plateau of Ecuador, following a gradient of remoteness, in order to again classify the type of diatom responses that climate and human activities, these changes induced in diatom assemblages. So the 4 lakes span the last 2 millennia, and we analyzed the diatom assemblages and geochemical content, the diatom calibration dataset, which is a subset of our wider tropical South American diatom database, which I'll be talking about later in a minute, comprises 125 lakes with environmental data associated in it from the northern Andes. We also use 4 different paleoclimatic records to provide a context in paleoclimatic changes and most importantly, variability of either climate conditions. So in terms of analysis, we analyzed trends of 2 types of beta diversity time series. One, the cumulative one, which is an indicator of the direction of change. So we measure from the baseline, in this case, the first observation in our sediment core, and the consecutive beta diversity as an indicator of the magnitude, so how strong diatom communities are changing over time. And we correlated this with drivers of climate, delta 018 records, and also human factors as reconstructed by archaeologically recorded radiocarbon time series and cropland areas. So these are the results. In this study, we collaborated closely with Albert Ruiz from the University of Berkeley and collaborators. And here I'm showing you the diatom beta diversity time series in the 4 top plots, which each represent one different lake. And at the bottom, we have the drivers, the cropland area, temperature, delta 018, and paleodemographic trends as reconstructed using archaeological time series. So anthropogenic impacts over the last few centuries accelerated and even synchronized cumulative diatom turnover in the less remote lakes such as Yaguarcocha and Yaviucu, and this is well exemplified by the coherence in the last assemblage level chains reconstructed using constrained cluster analysis, CONIS, for the last 150 years. Even in Lake Yaviucu, the cumulative diatom turnover showed a trend away clearly from the initial conditions. And also Lake Yaguarcocha, although it recovers thereafter, it also showed an increase in this latest diatom assemblage level transition. But a clear pattern is that in all lakes, the diatom cumulative densities return after each conic transition, and this is, and we interpret this as lakes may exhibit resilience to a variety of stressors because changes in paleoclimate conditions, such as the medieval climatic anomaly, drying conditions, and weather conditions following that, increase in anthropogenic impacts in lake catchment systems, and as well volcanic eruptions in Piñan and Fontococha lakes. And even though diatom, the diatom community do not follow a critical transition, except in Lake Yaviku, which I'll be exploring in a much more detailed analysis in a minute. A shift towards a landscape perspective of environmental changes, as you saw before in our previous results, is needed to assess whether or not changes are within the bounds of variability or changes are entering into new states. In a recent study collaborating with Melina Feidel and colleagues from the University of Cuenca in Ecuador, we analyzed 20 lakes in Ecuador to compare historical diatom assemblages before major anthropogenic changes happened, so more or less 150 years ago, before the Anthropocene with modern time. So we applied a Procrustes ordination technique between the two data set matrices representing 150 years ago and present time. And the longer the lines represent, the more dissimilar lakes are. So the ordination clearly separated two groups of lakes, the ones located in the Ecuadorian Interandean Plateau and the ones located in the high Andes, so in the Paramo ecosystem. Temporal trajectories are relatively stable for the vast majority of lakes, suggesting again, in this case, exemplifying well the shifting baseline concept, that natural or baseline conditions are might not be so different than the modern conditions. However, in a couple of lakes, specifically this one and another one, are clearly falling outside or changed completely from historical conditions, suggesting that they could be novel, or they might include novel communities, and so referring to natural baselines might have little or no relevance and might require reinterpretation. So a natural question arises now. So to what extent, how do we measure and by how much resilience is changing? While the study of resilience have been approached by different quantitative methods, very few attempts have been made, I believe, in linking theory with different multivariate statistical approaches when we deal with data that are rich in terms of species and community data, such as diatom paleolimnological observations. An heuristic approach, if you remember the ball and cap analogy and regime shift, would be to ask if the temporal trajectory of systems are outside the normal bounds of variation. So knowing to what extent ecosystems have traveled compared with the reference baseline, but for instance, in this scenario, return to initial conditions may help identify ecosystems that have recovered their initial condition and therefore they are resilient, versus ecosystems that might be falling outside their basin of attraction and might be crossing critical thresholds. Of course, these are not telling us the driver or by how much systems are changing its function and structure or suffering a regime shift, but it can pose question and hypotheses, temporal trajectories of ecosystems if we can compare a community with driver data. Quantitatively, also, we can model multivariate nonlinear trends of communities using more advanced statistical methods. For instance, GAMs, generalized additive models, are especially suited for modeling poorly behaved time series such as paleo, which are often characterized by uneven sampling, change in variance due to sediment compactation. So we need a statistical model that is able to learn from the data instead of preparing data to adjust into a model. So here I'm showing you an hierarchical generalizative model developed by Erik Pedersen and collaborators a couple of years ago, from which I've been the pleasure to work with, and I'm showing you our recent study applying this method. So the idea here would be to model all the species in the community, but allowing each individual ones have their own level of smoothness, while accounting also for a global smoother that is an indicative of the community level temporal trend. So in this way, we are trying to overcome the issue of dealing with multivariate data and sometimes reduce the variability using PCA and modeling the first axis of PCA, for instance, which is something that we are likely to throw away much of information included in the communities. When we have this method or the trends in the Diatom communities modeled, we can calculate average rate of change and see periods when communities are shifting rapidly and fluctuating around their regime. This is exactly what we did in our set of 4 lakes in Ecuador, collaborating closely with Gavin Simpson and Erik Pedersen, and we assessed abrupt changes in Diatom community data using this novel time series analysis, nonlinear time series analysis using hierarchical GAMs and rate of change analysis. Fluctuations of lakes, as you can see here, follow relatively stable periods, and interestingly, we can compare lakes that are geographically distant and limnologically distant as well, and observe synchrony, for instance, in the rate of change in Fondo Cocha and Yaguarcocha lakes following climate changes. Again, for Lake Lavircu, the one that we thought that follow a critical transition is again reinforced by the hierarchical GAM and the rate of change, in which we observed an abrupt decrease in the mean regions of diatom species versus the previous time were relatively stable. So the lake now is losing resilience and might be entering into a new alternative stable state. While this time series analysis is suited or can be used to see how much and when changes are following abrupt changes, they are not telling us about the drivers. Other multivariate ordination techniques such as PCA can help characterize the nature of change when coupled with GAM. So this is what we actually I'm showing you here. This is a combined GAM PCA analysis that allowed us to track fossil core trajectories as function of contemporary gradients of environmental change, in this case total phosphorus. So for instance, for Lake Piñan, which is one located in the high Andes of Ecuador, relatively far away from any human stressors and only following gradients of variability in climate, the temporal trajectory, as you can see, does not cross any threshold in total phosphorus. So we can use this temporal trajectory to say this lake is relatively stable within their bounds of variability. Again, going to Lake Yavircu, the one that are regarded as a model of potentially a lake crossing a critical threshold. In this case, the datum core trajectory indeed crosses several times these thresholds in total phosphorus, and therefore total phosphorus might be a driver that could be taken into account when looking at potential causes of critical transitions in this lake. A further step to infer drivers of change, if you remember the adaptation of the space for time substitution transfer function analysis in a previous slide, is to explore bivariate relationships among temporal predictions of diatom responses with expectations from spatial reference set of sites. This might allow us to generate multiple hypotheses of nonlinear changes and alternative stable states. For instance, here for Lake Yaviuco, again, we have three different types of communities as characterized by CONIes, or cluster constraint ordination techniques. And when we infer values from space and temporal expectations, we can make correlations about these two sets of matrices and assess their fit, right? So for instance, if we hypothesize that increase of productivity is the driver of change in Lake Yaviuco, total phosphorus versus siliceous titanium ratio, which is an indicator of past changes in productivity, we observe how the older datum community types behave similarly and very different from the most recent one. So there is a discontinuity happening here that can be another line of independent evidence that total phosphorus might be a proxy or might be the cause why these datum communities are changing or losing their resilience regarding the previous state in the ecosystems. In contrast, another hypothesis could be, okay, so the coupling between water temperature and past productivity in the lake clearly revolved in totally different directions, and this is a line of evidence that is telling us that the responses between productivity and warming of diatoms were uncoupled. So for the same rate of warming, a diatom would not be responding in the same way depending on the trophic state of the lake. A change in variance has been proposed as well as an indicator of ecosystem resilience, and that is, increase in variance may be indicative of an ecosystem approaching to a regime shift. So here we model variance of principal curves of diatoms, pollen, and pollen indicators of human impacts in Lake Yaviuco. This is a nice collaboration with Majoy Donastimiento and Mark Bush from Florida International University. Each component indicate different type of environmental condition and human components of the lake, and we modeled principal curves, which is an analogous of the first axis of PCA with GAMS, and we inferred variance as an indicator of a continuous trend in resilience loss. Variance trends of diatoms reinforce the resilience of communities in Lake Yaviuco because the increase and decreases of variance over time, so ups and downs. At the vegetation level, effects of climate change do not see a significant increase or decrease in variance. But again, this increase and decrease in variance is indicative, for instance, using the agropastoralism indicators or indicators that showed impacts on the lake catchment system, marked a clear increase when the Europeans colonized the South American continent. So it's a nice way to see impacts and loss of resilience if we can other paleontological records with this similar time series, and also then we can compare across components of the lake systems to see if aquatic changes are mediated by vegetation or human impacts, and infer changes in synchrony and dependencies between types of proxies. So for the last portion of my talk, I would like to draw your attention on the key aspect that is quantifying ecological data of indicator taxa. Nature is correlation, and given the intricate correlational relationships among local and regional variables, it is key to determine the geographical extent of sites and how well the ecological characteristics of taxa are quantified, considering emergent properties when we can really tease apart or model the shared contribution of variability of environment, climate, and space. This is, for the case of diatoms, different levels of higher biological organizations such as metacommunities and biogeography, considering niche and dispersal assembly processes are a nice example of an emergent macroecological properties. In the face of global change, combining macro ecology with more traditional local-based ecological assessment of diatoms is timely because habitat fragmentation and water degradation, all of these regarded as local processes, will likely scale up to catchment levels or larger regional scale spatial scales due to the increase of the interconnection of impacts such as warming, water abstraction, etc. And this is what this conceptual diagram wants to exemplify as a process is acting at different varied spatial and temporal scales. So central to test these ideas of emergent properties of macroecology in diatoms is our tropical South American diatom database. This database contains distribution and patterns of species across lentic and lotic environments, including predictors that describe local, so in lake and river and stream limnological conditions, and regional factors such as landscape variability, climate conditions, and also historical reconstructed human impacts in the basins. This database is actually being prepared for a publication, but this has been publicly available in GitHub through datasets and R scripts that allow the user to manipulate and visualize species distribution and environmental characteristics of the sites. So now we'd like to draw your attention on some examples and studies that we performed using this database. So we began asking at species levels what environmental factors drive plantic diatom taxa distributions, because planktonic diatoms are highly sensitive to thermal lake structure and how lakes respond to warming and nutrient conditions. We modeled a distribution of the most abundant plantic diatom taxa across the tropical Andes and the adjacent Amazon lowlands. And as you can see in this plot, we observed a broad elevational and latitudinal distribution, so no clear geographic restricted patterns in these diatoms. However, at species level, also different limnological controls correlated with several taxa. These are logistic generalizative model comparing presence absence of diatom taxa with different limnological variables associated with changes in the lake thermal structure. And as you can see, water temperature, for instance, we did not observe any clear pattern as this temperature being overarching variable affecting the atom distributions in our set of samples. At community level, we asked if regionalization of lakes, so to what extent environmental characteristics of the lakes and similarity among them can act as meta-community units for diatoms. So meta-communities are a set of local populations connected through dispersal by potentially interacting species. And we wanted to test these macroecological ideas on diatoms using our large-scale database. We began plotting into a non-metric multidimensional scale the distribution of the diatom data and associating them with lakes, with group of lakes as lake districts, so lakes that share similar environmental, climatic, and physical characteristics. And interestingly, we observed how these lakes were arranged, not only lakes in terms of environmental conditions, but also the datum community of these lakes and aquatic bodies arranged nicely following a latitudinal gradient, which at the same time integrates limnological variability. Also, lakes are arranged nicely along a landscape configuration which integrates variables such as percentage of aquatic habitat and complexity of the lake catchment system. So large-scale regional ones explained or were important in telling us diatom species distribution in tropical South America. When we analyzed all together using a variance partitioning approach, comparing different portions of variability explained by different sets of variables and see the proportion of variability explained by these different sets and the shared variability by groups of variables, we observe how geo-climatic and broad-scale variables outperform the role or the signal coming up from the local factors. So again, this was an independent line of evidence that diatoms are also responding to large-scale factors, and some emergent properties not explained by the local conditions are playing a role, and this needs to be taken into account when characterizing diatom indicator taxa for limnological approaches as well. Focusing on diatom species richness in different regions from tropical South America and along the Andean Cordillera and the adjacent Amazon lowlands show that diatom floras are regionally distinctive, so we observe a relatively low average proportion of shared diatom species among the studied regions and high levels of diatom turnover or biodiversity. Importantly, we also observe major differences between the highlands and the lowlands, and even among the regions, which suggest again the relevance of landscape configuration, altitudinal gradients, and the influence of both regional and local climate. In order to quantify that and get and shed light into what might be the factors explaining diatom species richness, we applied a generalized linear mixed model to understand variables that are more important in explaining diatom species richness following the ideas of island of biogeography approaches. So again, testing macroecological hypotheses on diatoms. The island of biogeography by MacArthur predicts that the species number will be an equilibrium, so colonization versus extinction rates is a function of habitat isolation and size. So we modeled diatom species richness using the same set of explanatory variables, combining local and regional factors, and interestingly, our results and linear models of diatom patterns indicate a significant increase in diatom species richness with increased water connectivity, which is the proportion of water bodies surrounding the study lakes or the water body of a study. And this suggests that a decrease in the remoteness of the system increases species number, which is one of the overarching hypotheses or fundamental ideas of this island of biogeography. I would like to finish my presentation with the most important take-home messages. Synthesizing data and diatom data at different temporal and spatial scales and methods might provide great new tools for estimating resilience through a lens of long-term limnology and assess what type of diatom community type responses climate and anthropogenic stressors induced on this highly important aquatic indicator that diatoms are. Estimating temporal trends in various metrics of paleolimnological time series is also key to understand when, where, and by how much acceleration of drivers in ecosystems caused by climate but also human impacts are important. Confronting spatial and temporal responses, so using a combined paleo-biogeography approach as a modification of the space for time transfer function method, is a promising approach to explore community alternative states, so uncouplings between the space and temporal expectations of the diatom communities to environmental change, in tropical lakes over time. Summarizing the diatom biogeographic patterns, regionally distinct floras with a significant association of geographic groups of lakes arose in our observations, in our results, primarily by determining latitudinal gradients of species richness and community structure, and also estimating the joint contribution of environmental filtering or niche-based assembly processes and dispersal dynamics affecting datom community composition. And perhaps more importantly, thinking about shifts, non-linearities, and baselines. Once we start thinking in those terms, we can start address global change problems such as historical baselines, the timing of inception in human impacts on aquatic communities, the trajectories, rates and magnitudes, and most importantly, their resilience, or to what extent ecosystems absorb disturbance without shifting without changing its function and structure. And until here, my presentation. Thank you very much for your attention, and I hope you all have wonderful and inspiring virtual ideas. These are my email and website, so don't hesitate to contact me if you have any questions. Thank you.